Wow, good morning. It's so lovely to see all of you logging in with us today for this great webinar, Innovations in Agriculture for STELR and the Victorian Challenge and Enrichment Series. Today we have Dr. Angela Vanderwaal with us, but firstly I would like to begin with our acknowledgement of country as we gather for this meeting from different places, prominently around Victoria. I'd like to acknowledge the traditional custodians of lands on which we meet. I'm joining you today from Ngunnawal country, which is in Canberra. But I pay our respect to elders past, present, and to any Aboriginal and Torres Strait Islander people who join us online today. As we share and discuss our knowledge and practices, we acknowledge the deep knowledge forever embedded in custodianship of country. And so today we are going to hear from an award-winning researcher. Angela has won an Agri-Food Award from us here at ATSI and has done some fantastic work in the agricultural sector, and you're going to hear a lot about what she's doing. But during Angela's talk, if there's anything you want to ask, please put it into the Q&A chat box, which is at the bottom of the screen, and we will get to the questions either during the talk or definitely afterwards. So we'll have a Q&A discussion at the end of her talk as well. So if you want to wait until then, please feel free to do so. Also, if there's any problems at all with your feed, you can ask us some questions and we may be able to help out. But we will be recording this session and you can access it after as well. So I will hand over to Angela now so she can tell you a bit about what she's doing.
Excellent. I will share my screen. So can you guys see my presentation okay, Camille?
I can indeed.
Beautiful. And can you see the mouse pointer?
Yep, everything's good.
Excellent. All right, we'll jump into it. So what I thought I would do today is, I guess, talk a little bit about who I am, where I work, what I do, and sort of how I got here. So feel free to, like Camille said, jump in and ask questions at any time. So I am a senior research scientist at the School of Biosciences at the University of Melbourne. I have been in my current role since 2005, so which is probably before some of you were even born, which is depressing. I am based here in Horsham, which is about 3 hours northwest of Melbourne, but I am all still heavily involved in my lab in Melbourne. So I have students based there, and pre-COVID times, I would be in Melbourne at least sort of a week every month. But now with COVID, they are all sort of online meetings, but that's the way it is. So whilst I work for the University of Melbourne, I'm based here at the Grains Innovation Park, which is run by Agriculture Victoria, so the State Department, and I'm affiliated with a private business called Marcroft's Grains Pathology. So they specialise in diseases of grains, in particular canola. And my colleague Steve, who runs the business, has been working on the same projects as me for a long time, which is why I made the move to Horsham back in 2013, just to make our project sort of seem together a little bit better. And it just made sense when canola is grown around the Horsham area to be based out here. So I thought I'd start by giving a bit of an overview of what canola is and the disease that I work on and why it's important before I sort of jump into, like I said, where I've come from and things like that. So canola is the pretty yellow crop that's out there flowering at the moment. I think it's the prettiest of the crops, but I might be a little bit biased. Back in 2017, 2018, we hit $2.1 billion worth for canola. So that was the biggest canola had been worth in Australia. So it's now the third largest crop that's grown for grains. The product that's of importance is the seed that is produced. So that's harvested and they extract the oil, which is used for obviously canola oil, which is used in cooking and things like that, but it's also used as a biofuel. And the leftover seed or the shell of the seed is used as a feed stock for cattle and sheep and things like that, so the meal. As well as being an important cash crop, so obviously you get paid per tonne of canola that you produce, it's also an important break crop. So you can't grow any cereal crop routinely in the same paddock. So if you grow wheat on wheat on wheat on wheat, then it has complications with disease and things like that. And the same if you go canola on canola on canola. So all these crops are rotated to reduce the disease and the control, to help control with weeds and manage nutrients. So it's got a range of important factors when it comes to the whole farm approach. So the thing that I work on is this thing called blackleg disease. So it's a fungal disease. It's caused by the fungus Leptosphaeria maculans. And this is just a range of the symptoms that you can see down here that it causes. So this disease is a stubble-borne disease. So what it means is that it lives in the stubble or the crop debris that's left at the end of the growing season. And each one of these black dots is a fruiting body. So here are the fruiting bodies here. And each one of these releases thousands of spores. And then when these spores land on the plants, they get in and cause these disease symptoms. So you've got here some seedlings and some leaves are infected. So you can see these big lesions here with the black dots in them. It then grows down into the crown or the base of the plant, and it colonizes the inside and the vascular tissue, and it blocks it. So it's like having a water pipe that's got junk in it and it gets blocked and your water can't move up and down the plant. What that means is obviously you can't get nutrients up to the top of the plant from the root system, and it causes those plants to wilt and die. It can also infect the upper parts of the plant. So here it's infected a flower that you can see there, and then it grows inside the stem. So this is up the top part of the plant, and again, it causes this blackening inside the plant and really blocks it up so it can't move the nutrients. And lastly, it can also land directly on the pods. So the pods are what are harvested at the end of the year with the seeds inside them that you can see here. But when you get these lesions directly on the pods, then all of a sudden the fungus can infect the seed directly underneath it, causing them to shrivel and go all dry, and then they won't actually germinate if you want to grow any more seed, and they won't produce any oil. So it has lots of different parts of the disease, and we do lots of work to try and control all of these different aspects. But the hardest thing about blackleg is because it's a stubble-borne disease, there's lots of it about. So this is a single paddock, and what you can see here is all of this rubbish that's left at the end of the season is the stubble. So each one of these pieces of stubble will look like this and produce thousands and thousands of spores. So essentially, you've got millions of spores out there every single year that you have to try and deal with if you're a grower. So it's a really difficult disease to control. You'll never get rid of it. It's always going to be around. So all the research we do is about giving growers opportunities to minimize the impact of the disease and try and do the best they can under the circumstances. So our research, we take what's called a genome to paddock approach to controlling blackleg. So this is an example of what happens when you don't control blackleg. So you can see here, every single plant has been infected and it has died and they've fallen over. And so you'd get no yield from this whatsoever. But when things go right, you can see here on the right, you've got some healthy plants there, all growing nicely, where there will still be blackleg present in these plants because the plants can tolerate a little bit of disease without any impact to yield. So if you do everything right or the best you can, you get this sort of thing happening. So the research that we do, it sort of encompasses all the different aspects that options that you have for controlling disease. So we can control disease through breeding for resistance, through cultural practices, and through fungicides. And our research looks at all three aspects in this to controlling the disease. So for breeding and resistance, we are identifying the resistance genes in the cultivars. We develop molecular markers for breeders to use to help breed for better resistance in the future. And then we also monitor these resistance genes in the field. And I'll talk a bit more about that in a minute. We've also done a lot of research to show that cultural practices can minimize disease. So we know that if you grow this year's canola crop at least 500 meters from last year's crop, it will help minimize the disease. And if you manage the stubble and try and reduce that amount of stubble that's left at the end of the year, that will also help. So we've just had a question come in that says, how long does blackleg take to come in? So what happens is, if I just go back to this slide with the stubble. So this will be what the paddock will look like at the end of the year. And what happens is that stubble will stay there. And then during the autumn months, when it starts to get cold and starts to rain, the stubble gets damp, and then the fungus starts to develop these fruiting bodies, these black dots. So it needs cold, wet weather. And that happens sort of from April, May until the start of June. And then every rainfall event from sort of the start of June through till September, these spores will be released. And so every single rainfall event, you'll get more spores being released from the stubble, which can then land on the plant and start to cause this level of disease. So once the spores land on the plant, it takes between 2 to 3 weeks for it to start to show the symptoms. And so every rainfall event, 2 to 3 weeks later, you'll start to see disease coming up. So it happens constantly throughout the year. The problem with that is, of course, that when you put your crop in, you also put your crop in at the same time when it's going to be raining, because you need that rain to get the seed to germinate. So unfortunately, you can't avoid the disease. We've got another question that says, if an animal eats the plants infected with blackleg, would that infect the animal? No. So this disease, the Bleptospherium maculans, the fungus, it is specific to Brassica species. So it can only infect Brassica. So your other brassicas are like mustards and turnips and broccoli. So it can only infect those kind of plants. It can't infect humans. Can blackleg affect a seedling that has been planted maybe a month ago? Yes, it can. So if you've got seedlings there, so if we go back to this page, if you've got at any stage during the development, if the fungus lands on the plant, it can get in there. So when it's a seedling, it'll get in on the cotyledons. When it's a leaf, it'll get in on the leaf. If there's flowers, it'll get in on the flowers. If there's pods, it'll get on the pods. So it doesn't matter what growth stage the plant is at, it can get in there. But what does matter is once it gets in there, it needs enough time to then grow and start causing all this black necrosis that you can see here. And that takes, well, actually, we're not quite sure how long that takes, but it takes about, we think about 3 months to start causing lots of damage. So these lesions only take 2 to 3 weeks to appear, but for the fungus to then grow down and into the stem, and it's the stem blockage that's the important part when it comes to yield, that takes about 3 months to occur because the fungus is only really slowly. No, it doesn't affect humans, Steve. Only, yeah, like I said, only the plants. So, which is lucky because it means I don't have to worry about anything when I'm out there working on it. So, back to the research that we do. So, like I said, we identify all these things for the resistance side of things. We also look at the stubble. And then we also have been involved in developing strategies for growers to use fungicides to minimize the disease. We also provide information all the time to growers about what sort of disease pressure there is. So when you've got a much wetter year, you get a lot more disease because, like I said, every year when you get that stubble getting wet, sorry, every rainfall event when you get that stubble getting wet, spores are released. If you don't have any rain, those spores can't be released, so you have a lot lower disease pressure. So we give advice to growers every year about whether they should be thinking about fungicides. And then we're also doing a lot of work around fungicide resistance because if you use fungicides, there has the potential for the fungus to be able to mutate and then no longer be controlled by these fungicides. So we do work in that field as well. So we do a range of different things. It keeps us very busy. But the main thing is that
Angela, we had a quick question. If you could just explain what a fungicide is.
Sure can. Okay. So, you know, if you guys get sick and you get a bacterial infection, the doctors will prescribe you antibiotics. That's a substance that then can then kill off the bacteria. Or fungicides are the exact same thing, but for fungi. So essentially it's a chemical that can get into the fungus and kill it and stop it from being able to grow. So it's usually you spray it onto the paddocks and then it will kill off the fungus that's there. And it works the same way as antibiotics do for us when we're looking at bacteria. So yes, in terms of our research, we are funded by growers' money. So what happens is every time a farmer harvests their crop, they get paid a certain amount of money for that crop, and 1% of that then goes into a pool of money, which is used for doing research to help them then increase their production on their paddocks. So this is through the Grains Research and Development Corporation. And so they then fund our research. So as a consequence, what this means is that the research that we do has to have industry output. So it has to have things that are relevant for the growers so that they can go and put those into practice on their farm and see the benefits of the research. So a few of the things that we do around that is that we release information to the growers about what their varieties are. So growers can put in, there's lots of different types of canola you can grow. And so we provide information about each one of them so that they can work out whether it's resistant to blackleg or it's not. And we've also developed a application, like an app that's available on tablets that growers can put in all the information about their farm and work out how much how much yield they're losing due to disease. And then they can change things what they're doing test things in this app to say, okay, well, if I now put a fungicide on, will that help me with my disease? So they can look at that and work out the best strategies for their farm. We also then provide this regional advice to growers. So every year we go out and we monitor the level of disease in all the different cultivars across the country. And we can then say to farmers, look, we think there's a problem happening in your region, and we can give advice to industry. So back in 2012, this was a really good example of when we did this. We found that these varieties that are called Group D looked like they were going to get lots of disease on the Eyre Peninsula in South Australia. And so we gave the farmers a warning to say, we think you guys should go and grow a different variety next year because we think your resistance is going to be broken down. And indeed, when we came back the following year, that's exactly what did happen. So this is what happened on the Eyre Peninsula. Our Group D variety was completely destroyed. And if you had have grown it, you wouldn't have got any yield. But all the farmers had listened to us and they had switched to these Group F cultivars. And so they got pretty much no disease. And we worked out that that saved those growers around $13 million for that 1 year in potential losses. And so as a researcher, to know that your research has that kind of impact is just amazing. And it makes it really enjoyable to be able to go out there and give that advice to growers and see the implications. So that is something that we do each year. We monitor these crops and give that information regularly to growers. So I thought I would switch tact now. So that's what the research we do is, but now I thought I would talk about a little bit more of the specifics of what I do. So I thought I'll talk a little bit about what my average week looks like and things like that to give you a bit of an idea of what science looks like on the ground. So essentially, this is an example of what I did last week. So I did some culturing of isolates. So we had the stubble that I showed you earlier produces one type of spore, but this fungus actually produces a second type of spore, which is called an asexual spore. And what it means is that they're actually all clones of each other. So this ooze here that you can see here, this pinky colored stuff, that will be millions and millions of the asexual spores. So they'll all be clones of each other, which makes our research very easy because what it means is we can take this ooze and put it in water, and then we can use those spores to infect plants, and we can repeat the experiments many, many times and do different things. And it's always with the exact same isolates, which is very cool. We use those to inoculate plants in the glasshouse. So this is just a little video of one of our glasshouses with all our plants in it. So last week I did some scoring of disease in there. The glasshouse is my favorite place to be because in summer it is nice and cool, and in winter it is lovely and toasty with the sun and the heater. So I always love being in the glasshouse. I also worked on a paper. So down the bottom here is a figure that I've been putting together looking at a range of different isolates with different genotypes. And also I was trying to do some troubleshooting with a molecular assay. We're not meant to have any of these blue dots up here. So trying to figure out what's going on. And also doing some reporting for report writing for a funding body. So, you know, our weeks do vary quite a bit, but that's what I was up to last week. And then I thought I would just sort of give you an overview of sort of how my roles change throughout the year. So essentially here I've got the clock with the months of the year on it, and depending on the time of year, I'm doing different things. So in February, I spend a lot of time doing industry updates. So we go and speak to a lot of growers and farmers and researchers and canola breeders about our research and give them the most up-to-date information about what we've been up to. Then in March and April, that's the time of the year when we put all our trials in the ground. So we do a lot of seed weighing and get thousands of packets of seeds ready and distributed to people to put them in the ground to sow our trials. And then sort of from sort of mid-May through to August, we start looking at all those trials that are in the ground. So that might look at seedling infection. And so we can go out and say, okay, we're seeing lots of disease this year. If we're seeing lots of disease, we'll be saying to growers, you might want to be considering to put a fungicide on. Or if we're not seeing any disease, we might say, look, guys, you won't need a fungicide this year, save your money. We do some experiments in the glass house, which is down here, where we can infect plants with different resistances to look at what's going on. We do an annual isolate collection. So that's where we take stubble from the field and we collect individual isolates and then we look at them and to see what's going on at the genetic level. We have to count our trials. For some of our trials, we've got lots of different types of them, but for our blackleg nurseries, which is one kind of trial, we have to go out and count them. It's always wet and cold and miserable, but I would argue that the team look pretty happy there regardless, or at least pretending to. Come August, we're looking at our flowers and looking at how much infection we've got for fungicide decisions for the growers again for that later infection. And then sort of from mid-October through to November, we're starting to travel around to all of our sites and look at how they're looking for crown canker. So we have 32 monitoring sites across the country, and we do all the monitoring in our Victorian and South Australia sites. And then we have colleagues that help us assess our New South Wales and WA sites. Then December, it's report writing generally and analysis of all our data. And then I'm very fortunate that I'm at a stage of my career where I then take all of January off and go on holidays with my dogs down to the beach because I'm a crazy dog lady. So that's sort of how my life changes throughout the year. We've also got experiments that are ran all year round. So we have glasshouse experiments where we look at either adult plants or we look at seedlings, and we also do lots of molecular stuff. So these sort of experiments are always running in the background. So I thought I would then sort of switch gear and sort of give you a bit of an overview of how I got here, because I think it's really important to realise that how we got here isn't always what you would expect and where we intended to go. So I grew up on a dairy farm. So this is me over here. Turns out I also thought I could be a hairdresser back then. I did try and cut my own hair, but I think it's a good thing that I didn't go down that path because I obviously wasn't very good at it. But because I grew up on a dairy farm, I've always had a passion for agriculture. And so that's one thing I have always known, that I wanted to work in some form of agriculture. And like many people that grow up on a dairy farm, I wanted to be a vet. So that was my goal for a long time as a child. And so back then, to get into vet, you had to go to uni and go into first year science. And then from there, you would transfer across into vet after you've done your first year. And so you had to go into your first year of science, do really well, and then transfer across. But the thing I found out when I first went to uni is that the uni is full of some really smart people. And it turns out I'm not up quite as high as I thought. And I just didn't get the grades to move across to vet. And so whilst I was a little bit crushed at the time, I thought, well, what I'll do is I'll do a second year and I'll transfer again at the end of the second year. And I guess that's when things really changed for me. So I had to pick my second year subjects, and I had done a little bit of genetics in first year, and I kind of did like it. So I went in and did genetics, and I fell in love with genetics, and that was mainly because of the lecturers I had. It's amazing how, and I'm sure you all know this, when you've got an amazing teacher, it has a huge impact on your enjoyment of a subject. And genetics was that for me. I had two lecturers, Dawn Gleeson and Len Kelly, who were amazing, and I just fell in love with genetics. And to be honest, I don't even remember whether I looked into vet after my second year of uni. I don't think I did. I think I fell in love with genetics and then decided to go on with that. And so I ended up doing a major in genetics. So I think 4 out of my 5 subjects were all genetic subjects. And then I went on and I did an honours year. So an honours year is a year of a small research project, as well as some lectures and stuff. Oh, someone's just asked me what made me fall in love with genetics. It was partly the teachers, and also I really enjoyed the human genetics side of things, which I guess I didn't expect, because like I said, I have this passion for agriculture, but I loved learning about, you know, how traits are passed on, what it means for, you know, if you've got blue eyes and your brother's got brown eyes, what that means and things like that. And I really enjoyed that aspect of it. And it probably helped that I was good at genetics. I think you always love a subject more if you are good at it. It makes it a lot easier. So I think it was a combination of those great lecturers and really enjoying and being, you know, good at understanding it that helped me fall in love with genetics. So when it comes to selecting my honours course, again, that passion for agriculture came out. And so I approached a academic that worked on insecticide resistance, and I did my honours year in the year 2000. That was probably the first time that I was scared as a young adult, in the sense that I had to be brave enough to go and approach someone who's clearly a very clever person and my lecturer, and I have to go and ask them if they would accept me into their program and be, you know, for those first steps into science. And it's a really intimidating thing to do, but I think it's really important to know that teachers and lecturers and all of that generally are in that area because they're passionate about it and they love it. So never be scared to go up and approach them and ask them for their opinion or their advice, because most of them really just want to encourage people to come in. So whilst it was scary, I don't think I've ever hesitated to go and ask people about what they do and what their projects involve ever since then. So I ended up staying in that lab and doing my PhD. So my PhD was 4 years. And again, that was on insecticide resistance. So I was working on the Australian sheep blowfly. So that's a massive problem for the sheep industry. It causes the fly-blown sheep. So that's where the larvae get infused into the bum of the sheep, and then they have to cut away the wool and the skin to get rid of the larvae or the maggots. And the only way to control it, if you don't do mulesing, is through insecticides. And so that then leads to the potential of insecticide resistance. And so I was looking at ways that insecticide resistance evolved and looking at that genetic side of things. So I was probably 6 months out from the end of my PhD, and I sort of started thinking, oh gosh, now I've got to try and get a job. What do I do? And I was very fortunate that a friend of mine who was about 6 months ahead in her PhD came up to me and said, oh, look, I just went and spoke to someone about a potential job that she has on offer, but it's not really something that I'd be interested in. I think that you would be, and perhaps you should go and talk to her. And so I went and spoke to this woman, Barb Howlett, about this job, and it was in canola, which I didn't really know anything about, and working on this fungal disease and trying to figure out the genes that are involved in disease of this pathogen. And she had interviewed for this job already, but she didn't find any suitable candidates. And we spoke about the job and what it involves and my background for about an hour and a half. And at the end of it, she offered me the job. And so I was very fortunate, and that's definitely not necessarily the norm of how you would get a job. But it was very fortunate for me. And so that's how I ended up where I am. And I haven't really left other than moving from Melbourne in 2013 to Horsham. So I do love my job. I'm very fortunate in that respect. Not everyone does love their job, but I do. And so I thought I would just cover the aspects of my job that I do love with you all, because I think it's really important. The first thing is the people. Science is just one big collaboration. You can't work in science in isolation. You always work with lots of people. So I manage 4 major projects involved in Blackleg, and each one of those projects involves collaboration. Some are here in Australia, some overseas. This is an example of one of my projects. So this is the main field-based project, and these are all the people that are involved across Australia. And some of these people have become some of my best friends, and you know, it's a really wonderful thing to be able to make these connections with people and work together to sort of look at the problem as a whole. And so I think that's a really fortunate thing about science. The other thing is the variation. So I put this slide up earlier showing you what my sort of year looks like. And I think it's so fantastic. Not all science is like that. I'm very fortunate that I work in agricultural research, so you have to work within the growing season, and so things do change regularly, which is awesome, and it keeps things interesting. I've just been asked how my work has been affected by COVID. The biggest thing that COVID has affected for me is the travel and making the collaboration hard. So normally we would all meet for meetings at least once or twice a year, which we haven't been able to do for the last 2 years. There's been no travel interstate, obviously. So this is Alex, my current boss in Melbourne. We have a number of students in Melbourne, which I haven't been able to see pretty much because COVID has restricted that. But in terms of my day-to-day running, I've been very fortunate that I've still been able to go into work every single day. I don't always, I do work from home if I've got a day of sitting at my computer, but if I'm got to be in the lab or in the glasshouse, we have been deemed as essential workers as part of the agricultural field. And so we have been very fortunate in that respect and have had very limited impact of COVID. It's a very different story for my colleagues in Melbourne. Because Melbourne has been in much stricter regulations than regional Victoria. They have had it a lot harder than we have, and there has been some impacts on our research there, but me personally, I haven't been overly affected, which is very fortunate. So the variation is really important. The other thing that I love about my job is working directly with industry and seeing my research being used. So I spoke about earlier how our research saved $13 million for growers. I mean, to see your research making those kind of differences to growers is just amazing. Our research features in local papers and industry pamphlets and things like that almost every couple of weeks, especially during the growing season. This month alone, I think I've given 5 presentations to growers and industry leaders. And so it's really nice when it's got that direct relationship to the on-farm. And not all my research is completely direct. I do a lot of work still in the lab, but knowing how that sort of fits into a story that leads down to that on-farm implications is really important for me. And it's the one thing I did know that I always wanted to be involved in when I went into science. The other big perk is travel. Not obviously during COVID, but under normal circumstances, there are conferences around the world. Like I said, you've got collaborations around the world. So I've personally got strong collaborations in Canada, the UK, France and Germany, which previously I've been to, and under normal circumstances I would go to probably at least once a year. I would travel internationally. There's also such opportunities to go and work in other labs. So I did a 3-month stint at a lab in the UK. I did a 2-week stint in a lab in France. There's always opportunities for exchanges. I've currently got a grant that is to fund one of my students to go over to Germany for 6 months. So when COVID allows, he'll be doing that. So the travel is really important. Someone's just asked me whether Blackleg has any pros. Well, it has pros for me because it gives me a job. But in terms of for growers, no, I don't think there's no advantage that I could think of that Blackleg would have over, yeah, I don't think there would be any advantages to having Blackleg in your crops whatsoever, I don't think. I've never been asked that before. Very interesting question. Is Blackleg a global disease or is it only present in Australia? It is actually a global disease, but it is the worst in Australia compared to everywhere else. So I should say it's a global disease with the exception of China. So China don't have Blackleg disease that's caused by Leptosphaeria maculans. They do have a similar form of the disease, which is caused by a very closely related fungus, but it doesn't cause the same limitations as the Leptosphaeria maculans Blackleg disease does. The rest of the world do have Blackleg. However, it's not as extreme as in Australia. And there's a few reasons for that. The main reason for that is that in Australia, we grow our crops over the winter months And that's when all the spores are being released. And so it's a high disease pressure situation. And the reason we have to do that is because our spring and our summer is so dry. So we just can't grow our crops over spring and summer. Whereas in Canada, they put their crops in at this end of winter, start of spring. And then because all that snow is melting and stuff like that, they have lots of water. And so then it grows over the spring and summer months and they harvest in summer. And it's similar in Europe. So it's because of the way we have to grow canola here to accommodate our rainfall events and things like that, it makes blackleg much worse here than the rest of the world. But it does mean that we are the world leaders in blackleg research. So that's a bonus, I guess, especially for me. Now, I did say I love my job, but of course, with every job, there does come with parts of it that you don't like. And these are the things that I don't like about science. So you have to do report writing, and report writing is so boring. And I'm sure you all appreciate that when you have to sit down and write assignments. Sitting at a computer is always challenging. Having said that, when you do put a report together and it shows you how much work you've been doing, that is always quite rewarding. So it does still have a little bit of a plus. One of the hardest things about science is failure. With experiments, they don't always work first time. They always need tweaking, and that can get demoralizing. But when they do work, they're amazing. So, but it does require a kind of, a typical, a particular type of mindset to be able to deal with those failures when they do happen, especially when you're talking about experiments that run in a growing season. When you put them in the ground in June and you don't get a result to November, if it doesn't work, that's 6 months gone and you can't do it again until the next year. So it can be quite difficult to deal with sometimes. And the other thing is finding research money. So when you start out in science, this is not something that falls on your shoulders. It falls on the shoulders of people above you. But when you get to the level that I'm at, for example, you have to then start finding the money for your research. So you've got to put in grant applications. Sometimes these are small, sometimes these are big, sometimes they're successful, sometimes they're not. I recently found out that I put in a grant for a 108-page grant, and I missed out on the money. And it was because of COVID. They cut the funding by 50%. So they only funded half the number of grants that they would normally, and they informed me that I missed out by 10%. And in a normal year, I would have got funded. So it can be demoralizing as well sometimes. I just got the question, do you work with the Department of Agriculture and Government, or is the research you do privatised? So we do work with the department sometimes. So our research is funded by the Grains Research and Development Corporation. So that's the money that comes from growers, which is matched by the government, and it's a big pool of money. And then you apply for that money to do your research. And so it's not privatized, but it's also not purely government. It's the body that sort of runs it is the Grains Research and Development Corporation, that sort of comes under a little bit of a blurred sort of in between those two things, if that makes sense. But we do collaborate with researchers from the Department of Agriculture here in Victoria. We collaborate with the Department of Agriculture in New South Wales and WA as well. How long does the experiments take to run? It depends on the experiments. So if we're doing field trials, they will usually go in the ground inside a sort of mid-May through to mid-June, and then we would be scoring them at different times during the year, and then their final assessment and their yields would be taken in mid-October /November. But other experiments like molecular experiments, they sometimes only take an hour to run the molecular experiments, and you get an answer. So it depends on what experiments you're doing. Our glasshouse experiments, for example, usually a glasshouse experiment where we're looking at seedling infection, we would sow the plants, and then 7 days later we can come back in and infect them, and then it takes 2 weeks for that disease to develop, and then we can score them. So you're sort of talking 3 to 4 weeks to run a glasshouse experiment. So it really depends on the experiments. Lastly, I just wanted to finish with a few life tips. I do get the opportunity to mentor a lot of people, which I love, absolutely love. And so these are kind of the things that I'm always telling people, which I think are important. So I think it's really important to have high goals, but don't ever be disappointed if you don't reach them. Like I said, if you had have asked me when I was 18 what I would be doing in the future, I would have told you I was going to be a vet. And whilst I'm sure I was disappointed at the time when I didn't get in at the end of first year, I have plenty of vet friends now because I'm a crazy dog lady, so I've got to have vets as friends to save me money. And to be honest, I would have had no desire to be a vet anymore. I think they have a very hard job and it's very difficult. And I do wonder whether I would have actually been suited to be a vet. So I'm very happy with where I got, but I think it's important to have the high goals, because if you have the high goals, it will help you to get to places that wherever that might be that you're going. So I'm very grateful for having that goal, but I'm not disappointed that I didn't get there. I think it's really important to take all the opportunities that are offered to you. So experience, helps you know what you want to do. So if you go out and you do your work experience and you go, oh my goodness, this is awful. Well, that's really important to know now before you go and do 3 years at uni and go and get a job. But it also gives you an advantage of getting jobs. Unfortunately, it's a bit of a double-edged sword when you start applying for jobs. People want you to have experience, but to get experience, you need to have a job. So it's a hard one. The other thing is that contacts are really important. So it's like I said, the reason I got this job was not because of anything other than pure luck and knowing the right people. And so that isn't always the case. I'm not saying that is always the case, but having those contacts are really important. And the other thing is to start writing your CV now. So I keep a record of everything I do. So what I've done today will go on my CV. It's really important to keep a record, because one day you might be applying for a job where you might need to have had some weird experience that if you've got it written on your CV, you can then use. So an example of that is that many, many, many years ago, I gave a presentation to my friend's prep class. And I really loved doing it, and I put it on my CV. And I then applied for a grant that needed to have industry engagement with the community. And so I then could come back and use that piece of information and say, this is one of the things I've done. Someone's just asked me what a CV is, and I didn't even think that you might not know that. So a resume is another word for it, but essentially it's a document that lists all of your experiences and your achievements. So you would put on there, so if when you do go to uni, you would say that you've got a Bachelor of Science, or you've got a Bachelor of Agricultural Science, or whatever. It would say whether you finished VCE, if you got any awards while you're at school, you would have that on your CV and things like that. And so it's really important to keep these things as a record, because you never remember them. You will forget. So have a CV and just add to that document all the time whenever you do anything. And then when you start applying for jobs, you can pull out the bits that are relevant to you. And I think you should always try and find something you're passionate about, because you spend a lot of time at work, and so you want to enjoy it. And I think that makes my job very easy, because I do love Blackleg. I did just get a couple of questions there. So I've got, how was Blackleg discovered? That's a good question. So Blackleg was first identified in Australia back in the 70s. It destroyed the canola industry back then. So it completely wiped out the entire crops, and they actually stopped growing canola until the late 1990s, when they discovered that there was resistance in some varieties. And so they reintroduced them and started doing it that way. But how it was discovered, I don't know. I have no idea. To be honest, I assume someone found some leaves with these sort of lesions on it that you can see here, and would have taken it back to the lab and grown out the fungus and started looking at it. Bo's just asked me, what double degree would you recommend with agriculture, please? Well, I think that would completely depend on what you want to do. If you want to do pure science, then I think you could just do your agriculture, or you could combine it with science. If you want to go out and become like an agronomist and things like that, then you could look at combining it with sort of economics and stuff like that, so that you have a good understanding of how whole farm systems work and things like that. So I think it would completely depend on where you're thinking of trying to head with your degree. If you wanted to go down like breeding, the breeding realm where you're breeding for varieties and stuff like that, then you might want to combine it with things like genetics, so in pure science. Yeah, so I think it just depends a little bit on where you want to take your degree in with that respect. So I think that's it for me in terms of a presentation. I've loved all the questions. If anyone's got any other questions, feel free to keep asking them because we've got a little bit of time left.
Yeah, we've had some great questions come up today. It's been fantastic. I had a couple of questions for you.
Yeah.
Just in terms of the working and the collaboration with people, is it very much, you talk about exchanging things, but is it just an exchange of ideas in some cases, or are there projects you would get to work with a variety of different people across? Yeah,
it's sort of a bit of both. Sometimes your collaboration is just exchanging material or isolates or things like that, but often it's actually experiments. So for us, for example, we have these trials across the country, and so we'll have collaborators in WA that will assess all the trials and send us the data, and then we all meet to discuss what the data means, and then they will extend the information to industry in WA rather than us go over there and give talks. They'll give the talks. It might mean that we do part of an experiment and then we need someone else who has expertise in a particular field that will then do the next part of that experiment and things like that. So it really depends on sort of what you're doing. But it's really a great way to increase your knowledge base as well, and really, get to go and share those ideas and have those discussions and stuff.
Yeah. I saw on your kind of year spreadsheet of things that you did to do, which is fantastic. It's great, like a plant cycle. You had a few graphing and things that looked like there were lots of computer work there.
Yes. That's one of the joys of becoming the senior research scientist, is unfortunately you get to spend more time at your computer than out in the doing the actual work. And that's a reality of moving, up the food chain. I now spend a lot of time. I've got, I think, 4 PhD students. We've got, based here in Horsham, we have 3 staff members. And so, you're managing their time and making sure they're doing the right aspects of the job. And, my role is to make sure we're delivering on all the aspects of our project and that everything's coming together, and then writing those reports for industry. And a big part of science is publishing your work. So you have to write manuscripts that describe a research project or a particular bunch of experiments, and then that goes and gets peer reviewed by other scientists, and then it gets published in a journal. And so one of your sort of quotas for science is publishing, and so I do spend a lot of time writing those up as well. In fact, that's what I was doing this morning, was writing up a paper. So yeah, I do spend a fair bit of time at my computer these days, but in the early days you definitely don't, but as you progress through, that becomes part of your role. But yeah, that's responsibility for you.
Well, it's definitely a good indication to our students to do those science reports that the teachers want you to write.
That's right.
Aims and conclusions and everything. It's good practice.
Yeah, and the thing is, it is actually what you end up doing if you own science. It's so yeah, I spend a lot of time saying things like that.
I always kind of ask people who come on our talks, what do you do when you're not doing science? You've mentioned being a crazy dog lady, which personally I'm on board.
I am a crazy dog lady. I have 3 Labradors, and I've also done a degree, not a degree, what do you call it? Not a diploma either, but anyway, I'm a professional dog trainer as well. So I run the Horsham Dog Obedience Club, and I train my Labradors. So they've 2 of them have got more titles than I do. I don't know if that's embarrassing or not. So I do compete with my dogs in obedience. And I think work-life balance is a really important thing, but it's your idea of work-life balance. When I lived in Melbourne, I used to work 12, 13 hours a day, but that was my choice. And because I love it, no one was forcing me to do those kind of hours. And so to me, that still was work-life balance, because I'd still go home, go and play my sport, do all those things. Now I've, a bit more senior and I've got my dogs. I do work my day is, I normally start work at 7:30 and I finish work at 4:30. And occasionally I work in the evenings or on the weekend, but not very often anymore. And I spend most of that time with my dogs and great friends and drinking wine.
That sounds some very great time to do when you're not doing science. I'm on board for that.
Excellent.
But I really loved your final comment where you said find something you're passionate about and just do that. And, not all of that stuff needs the degrees or anything. It's just that thing that you love, and you don't feel like work at all. So.
Yeah, I'm one of 5 kids, and we have all managed to find jobs that we love. So one's a builder, one's a diesel mechanic, one's a gardener, and one's works in early childhood. And it's taken us all of us have worked all over the world. One of my brothers has done a stint in Antarctica, and I think you don't always need a degree. It's about finding something you're good at and something you love. It makes a big difference.
It certainly does. And we've all really enjoyed listening to you talk about your work today and what you love and how you're helping cropping and farmers and the food of the future, which is really great. So thank you so much for being here, and thank you to all of our attendees and our question askers.
Yes, thank you so much for the great questions. Makes it so much nicer.
It does. And I'm going to just quickly chuck in the chat a link to our survey that I would really love if you would be able to fill it out for us. Let us know what was great about the talk and what you would like included in ones in the future. We'll also send it around as an email afterwards if you can't get the link out of the chat. But yes, we'll leave everybody there today, and thank you so much for joining us all here today.
Thanks, everyone.